Hey guys, this is Transcription and Translation, Chapter 17. So we have to look at Beadle and Tatum. These guys came up with the idea of one gene, one protein. What that means is for every protein has a particular gene that codes for it. Nobody really knew that, and so these guys performed experiments with mold, both a wild type, what you would find in the wild, and some mutants that were unable to synthesize the amino acid arginine. They determined from their experiments that each different mutant had a different block in the pathway, if you remember metabolic pathways, for making the arginine. So they had different enzymes that were not functional. You could bypass this pathway by adding the essential amino acid, the one that was missing. Here's a diagram. So if you look, there was an arginine 1 mutant, arginine 2 mutant, and arginine 3 mutant. And if they put in the, for example, with the arginine 1 mutant, if they put in all of the amino acids with minimal media, which means it has really not a whole lot, it can grow if it was given the precursors for the next amino acid in the pathway. Mutant arginine 2 could grow only if it was given citrulline, but not if it was given ornithine. And so that meant that there must have been a problem between ornithine and citrulline in the pathway. So the enzyme must have been non-functional. An arginine 3 mutant, it can grow only if it was given arginine itself. So they can't. If you look at the bottom, you see the pathway with the precursor molecule, enzyme 1, enzyme 2, and enzyme 3, and they were able to determine with this experiment that each gene coded for a different enzyme, which was a protein, and so therefore, let's move on. So, how do you get from one gene to one protein? You have to go through the processes of transcription and translation. And you probably recall that transcription is creating messenger RNA or mRNA from the coding DNA that's in the nucleus. And this messenger RNA is a disposable copy, if you will. It will be sent out of the nucleus to the ribosome. At that time, it will go through a process of translation, which is converting the messenger RNA into a protein with the help of transfer RNA. You need to recall that there's a triplet code, and that every 3 bases in your coding DNA will code for one amino acid, and it is called a codon. There are codons that exist for start and stop, and some amino acids have more than one codon, which is sometimes there's one that's preferred in a different organism, and the third base is often the one that is different in the codons, and it's been called the wobble base because it doesn't appear to be critical in the process, and it's the most likely one to change. You'll still get the right amino acid. So you must read it in the correct reading frame and the correct groups of 3, and if you're off, you'll get some big mistakes. And there's this molecule on your screen is tRNA. It's shaped like a T, which is really a good, easy way to remember it. But it carries an amino acid at the top end, you can see. And at the bottom, it has an anticodon, which will base pair with the codon in the messenger RNA. So, what's going to make a cell go through transcription? There's an outside factor, and if you recall from cell signaling, you'll get some kind of cascade, and that will cause a transcription factor to be made. And then this factor is going to bind to what is called the promoter region in the DNA, and it's a TATA box, and it literally is the base is TA. And this will allow for RNA polymerase. RNA polymerase is an enzyme that's going to catalyze the production of the RNA, and it will bind to the promoter and initiate transcription. So this diagram, then you can see the TATA box right above the words core promoter. There's an enhancer region further upstream. We'll talk about that later. And you can see that the RNA polymerase II can bind in this complex and begin making the new RNA. The TATA box at the start of the promoter region is approximately 25 bases away from the actual start codon. And so this transcription initiation complex, which is composed of transcription factors, RNA polymerase, the promoter, it's actually going to fold and move into place. And at the end of the transcription, when you're done, there is a terminator or stop sequence to tell you when you're done. So when you're done making this messenger RNA, it's actually a pre-mRNA in eukaryotic cells. There's a little bit of work, modifications that have to be done to get your final product. The first thing is called a 5' cap, and this is made of guanine bases, and you can see in the diagram it's in yellow at the far left. This is going to protect your RNA from degradation when it leaves the nucleus, and it has an attachment signal so that it can find its way to the ribosome where it's going. The other feature is the poly-A tail. It's from 30 to 200 adenines are added to the 3' end of the sequence. This will also protect the RNA from degradation, and it will help it to actually exit the nucleus. And the poly-A tail, as you can see, is at the 3' end on the right-hand side of the diagram. Well, we're not done yet. We have to do something called RNA splicing. We're going to cut out the non-coding regions, and these are called introns. Introns are intervening sequences in the DNA or RNA, and the exons are the actual coding regions. So you have these SNRNPs or SNRPs that recognize the RNA sequence for cutting, and they will cut out the portions that we don't want. Now, it's important to note that introns may be prehistoric coding sequence that we don't use. They may help in crossing over. If you remember crossing over, you've got to actually kind of cut the DNA and swap it, and so you can imagine things that would be important you wouldn't want to cut in the middle of, so it does aid in that. And it's also important in alternate splicing so that you can have different forms of the same thing, and we will see that later when we study antibodies later this year. So in this diagram you can see the exons are in blue, the introns are in white. And so when we first copy our first pre-mRNA in green here, the green and white, it's the whole thing. But we do cut out the introns, so in step 3 you can see the introns have been removed, and that's what's actually going to leave the nucleus for making proteins at the ribosome. There's something called the spliceosome, the term that's been coined, and that contains your SNRPs, those little cutting molecules, some proteins, and it will cut out your introns and join the exons. That's important. They have to be put back together, and that's what splicing means. And then there's also an RNA enzyme called the ribozyme involved in this process. Once we've done our transcription, we got to the ribosome, we have to do translation. We have to change the language from DNA and RNA in nucleic acid language into protein language or a polypeptide. So we're going to need some ribosomal RNA subunits, rRNA. Those are 2 pieces. They actually don't stay together. They only come together for the process of making the protein, and then they will separate. We need a tRNA that's carrying the amino acid to the ribosome so that we can put together our polypeptide, and we need that messenger RNA template. Each tRNA has its own enzyme, and it is shaped like a T, as we said before. And one end has an amino acid binding site, and the other end, at the hairpin turn at the bottom, has a 3' anti-codon. And the wobble base can bind nonspecifically. Again, here's our picture of our tRNA. At the bottom, you can see the codon-anti-codon pairing, and at the top, you can see the amino acid. Important to note that you can see, actually, they put a nice little green dotted box around the carboxyl group, and you can see your amine group. They are important. So how are you going to initiate translation? How are you going to get it started? Well, the messenger RNA will bind to this small rRNA subunit. There's a small and a large subunit, and this is assisted by that 5' cap that we looked at. the TNA with the start codon, AUG, or anticodon rather, will bind to the start codon, and this one has the amino acid methionine attached to it. After that happens, the large RNA subunit will then attach, and now you have a complete ribosome ready to go. At that point, the RNA ribosome will catalyze a peptide bond between two amino acids. The growing chain is moved to the A site, that's the acyl site, and the tRNA will move to the P site, the peptididyl site. The P site tRNA will move to the exit site. So we're going to leave behind our growing polypeptide, and the tRNA has to leave, so it goes to the exit site, and it will be released without the amino acid. In this diagram, you can see a little bit better. We have our three sites. So our single amino acid comes in at the first site. It's going to move then. It's going to the polypeptide is going to be handed off, and we get a growing chain, and then we go to the exit site, and the tRNA, as you can see, is leaving on the far left to go get reloaded, if you will. So this process will continue until it reaches a stop codon, and there is a release factor protein that will move into the A site, and it will cause the release of the polypeptide since it's not carrying any amino acids. At that time, the ribosomal subunits will separate, and then our proteins will go where they need to. And in case of membrane or secreted proteins, they will have a special signal peptide that will allow them to attach to the endoplasmic reticulum because they need to get outside of the cell. Once the tRNA is released from the ribosome, it must find more amino acids, and that's called tRNA reloading. And there's an enzyme called amino-acyl tRNA synthetase, and it will catalyze the binding of tRNA to its specific amino acid. And that is the end of the show.